we will be talking about completely about glycolipid. The glycolipid is a sphingolipid. All sphingolipids are glycolipid and all glycolipids are sphingolipid. So we will be talking about this sphingolipid, okay, about glycolipid. So in this sphingolipid, we will first talk about chemistry and function of sphingolipid. Then we will talk about disorder. This disorder are very, very high yield disorder for USMLE, for NBME. So you can see we have 6 disorders. 6 disorders. Among the 6 disorders, you can see these 2 disorders. They are very related. They are related. Actually, they are not related, but this presentation wise they are related and they are different in something. So we will do compare and contrast as well. And these are neuronal. So neuronal manifestation you will see in this 2 disorders. They are extra neuronal manifestation you will see in this 2 disorders. Predominant, if I telling about neuronal and extra neuronal, means predominantly it is related to nervous system and it is predominantly related to extra nervous system, neuronal. And then these 2 are completely related to nervous system, where you will see leukodystrophy. What is leukodystrophy? We will talk about this. I have already, actually, if you can remember, leuko means, and I don't know if I told you or not. Leuko means white matter. White matter. The white matter of dystrophy means degeneration. So white matter degeneration. So white matter degeneration disorder will have are these 2: Krabbe disease and metachromatic leukodystrophy. So this 6 disorder we will talk. We'll try to talk. This is 2nd class. Among this 13 class, you can see sphingolipidosis. So you can see 3 types of lipid. So today we are going to talk about this glycolipid. Glycolipid is present in outer part of membrane. You can see glycan. So glycolipid is always present in outer leaflet of membrane. Okay, these are all glycolipid. There is one sphingolipid which is phospholipid. So we will be also talking about this. Okay, so you can see these are the types of sphingolipids. These are the types of sphingolipids. You can see 1, 2, 3, 4, 5 types of sphingolipids. First is sphingophospholipid. This is not glycolipid. First is sphingophospholipid. These all are present in all these sphingolipid, even phospholipid. They are present where? Outer leaflet, not inner leaflet. They are all present in outer leaflet. Okay. So the first lipid you will see is sphingolipid. And here X will replace this. You can see this. This is sphingolipid. So this is phosphosphingolipid is sphingomyelin. Now second lipid is glycolipid. When this glycolipid is glucose containing glycolipid, only glucose bind here. I told you the compound lipid becomes compound lipid only because of at 3rd position, this position, a different group will attach. So this glycolipid will have glucose. If glucose is bound here, this is called glucoside. This molecule, you know, ceramide. This is called ceramide. Sphingosine plus fatty acid. Sphingosine. This is sphingosine backbone with fatty acid is ceramide. So in ceramide, if glucose is attached, this is called glucosyl ceramide. The other name is glucocerebroside. So these are other name, glucocerebroside. Same way, if in place of glucose, galactose binds, this is called galactosyl ceramide. Other name is galactocerebroside. Just remember galacto at least. You can add ceramide or cerebroside, no problem. Even if you have ceramide, it will be the same molecule. If you add cerebroside, it is the same molecule. So glucosyl ceramide and this is galactosyl ceramide. Now there are 2 other types of glycolipid which has oligosaccharide. One is globoside. You add globe here and it becomes globoside. You can remember like that. So globoside is, you can see glucose and 2 galactose like this. This is trisaccharide, is galactose. So this is globoside. This is what is called globoside. This is globoside. You can see this is globoside. You can remember this globoside like fiber. I will tell you the significance. So it is related to a disease that is called Fabry disease. So globoside, glucose, galactose, galactose. This is called globotrioside. This is called globotrioside. You can call it globotrioside because trisaccharide is there. That is why. Now, last but not the least, the derived name from ganglia, and this is called ganglioside. You can see this ganglioside also have glucose, galactose, galactose. But last galactose, if you compare with this, this is also galactose. But with the last galactose, this galactose is galactose amine. Okay, galactose amine plus galactose, 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 galactose. So this 4 type of oligosaccharide. With this, there is one neuraminic acid. This neuraminic acid is here is, you can I call it gangster. This is gangster here. Why? If it is not there, neuraminic acid is not there, it becomes globoside. If neuraminic acid is there, it becomes ganglioside. So difference between globoside and ganglioside is this neuraminic acid. This is a very important concept. Neuraminic acid is the difference between globoside and ganglioside. Neuraminic acid is the difference between globoside and ganglioside. So you can see this neuraminic acid is here. This is called ganglioside. Any oligosaccharide containing neuraminic acid is called ganglioside. Ganglioside, as the name suggests, it is present in ganglia, central nervous system, peripheral nervous system. So you can see the location also. Location is very important. If you understand the location, so now I can write, if you understand the location, you will understand disorder. For example, sphingomyelin present everywhere, both in neuronal tissue and extraneuronal tissue. And this glucose is a very common molecule. So you can correlate that way. This common molecule, because glucose is common molecule, that is why it is not a special molecule. That is why it is present in extraneuronal tissue. Galactose is a very special type of molecule. Okay, it is present in milk only. Milk is special. So it is present in a special tissue. It will form a special tissue that is called myelin. So galactosylceramide is part of myelin. That is how you can remember. Galactose is special and myelin is special. That is how galactose is present in myelin. So you can relate this galactose with myelin, galactosylceramide with myelin. Glucosylceramide, extraneuronal tissue in normal membrane, other membrane of macrophages, membrane of other cells, glucosylceramide is present. Now, globosyle is mainly present also in extraneuronal tissue, and it is also present in peripheral nervous tissue, not in central nervous system. Okay, it is not present in central nervous tissue. And this ganglioside is present in neuronal tissue, both peripheral. When I tell you neuronal, it is present both in peripheral and central nervous system. So see, these are predominant. It is not sharp distinction that it will always, some lipid are present only in extraneuronal. It is not present in the central nervous system. It is not like that. It is predominantly. So if you relate this, if you understand the location, predominant location, so you will understand what? You will understand the disorder. You will understand the disorder. So will you memorize? Glucosylceramide present where? Yes. Glucosylceramide present here.
Extraneuronal.
Yes. Glucosylceramide, glucose or glucocerebroside, extraneuronal tissue. Galactosyl is forming what? Or galactocerebroside?
Myelin.
Very good. Myelin. Galactose, special tissue. Galactose is special. It is present in a special type of drink, milk. So glucosyl, galactosyl. Then Next come globoside. Globoside present where?
Extraneuronal and peripheral neuron.
Very good. Extraneuronal and peripheral neuron. Peripheral neuron. And ganglioside.
Neuron.
Very good. Neuron. And then sphingolipid, that is sphingomyelin, is present everywhere. Okay, both. So if you remember 1, 2, 3, 4, 5, you will remember all the disorder. You will remember all the disorder.